Bio. This is Mr. B. We're continuing on chapter 12. We're in section 2 today finally, and we have that this section is titled Chromosomes and DNA Replication. And so we're going to get right in the first part being chromosomes. So first off, we're going to talk about what DNA and chromosomes are like in prokaryotic cells. So if you remember, prokaryotic cells are things like bacteria, things that they're the more simple types of cells that don't have all those organelles that we discussed. And their DNA is going to be located just floating around in the cytoplasm. So their DNA in prokaryotic cells just kind of floats around. And most prokaryotes have a single DNA molecule that contains nearly all of the cell's genetic information. So if we'll go and look at kind of a zoomed up image of what this might look like. So we have a picture on the left is a bacteria known as E. coli, and you can see it has its one single chromosome, just one strand, and then if we zoom in on it, we have what all those bases that we talked about. If you remember, A goes with T and C goes with G, and that is the same principle for prokaryotes as well as eukaryotes. It's just in prokaryotes we don't have that double helix that we said we did, that Watson and Crick came up with the help of Rosalind Franklin. These are just a single strand because they're more simple. So that's about all we're going to say for prokaryotes for now. So now we're going to look at the chromosomes of eukaryotes. So many eukaryotes have 1,000 times the amount of DNA as prokaryotes. So we've said we're kind of more complex than prokaryotes, and that's kind of what it shows there. Remember, eukaryotic are us, plants, fungi, things called protists. We've talked about all those before. And then eukaryotic DNA is going to be located in the cell nucleus inside of chromosomes. So I think we've discussed this before. We all know this. So eukaryotic DNA is located in the cell nucleus inside chromosomes, and our number of chromosomes is going to vary widely from one species to the next. Remember, we said fruit flies had 8, humans we have 23, goldfish actually have 40-something for some reason. So it just depends on what species you are. It's going to depend on how many chromosomes we have. So that all should be reviewed. And then some chromosome structure. Eukaryotic chromosomes contain DNA and protein that are tightly packed together to form chromatin. That's not a new word. We've talked about chromatin before. So chromatin is just the tightly packed DNA and protein. And then chromatin consists of DNA tightly coiled around protein called histones. So histones are a new word. They are important. They are the protein that kind of keeps chromatin held together. So the protein and the DNA wrap around the histones, and that keeps everything from coming apart. And DNA and histone molecules form nucleosomes, so that's another new word there. So when we just have DNA and histone together, we have nucleosomes. So when it's wrapped around, it's known as a nucleosome, and they pack together that form a thick fiber that will eventually make up our chromosome. So kind of a picture of what this looks like. So we have our biggest part is a chromosome. Zoom in on a chromosome, we have what are known as supercoils. And we go zoom in even farther of the supercoils, then we have a coil. We can see we just have our chromatin wrapped around these pink blobs, and these pink blobs again being our histones. And we can see right here we have several histones with the DNA wrapped around it, and that's going to form our nucleosome. And we go in even farther, zoom in even farther, we have our DNA double helix. So this is a pretty important picture, and kind of shows us the exact structure of our eukaryotic chromosomes. So the next thing we're going to talk about, since we kind of know what our DNA is made up of, now we're going to talk about something called DNA replication. And the word replicate just means kind of make another copy of, so it's going to be making a new copy of our DNA. So each strand of the DNA double helix has all the information needed to reconstruct or remake, like we said replication means, the other half by the mechanism of base pairing. Remember, we talked about base pairing last time as the A's and the T's and the C's and the G's matching up. And in most prokaryotes, so DNA replication begins at a single point and will continue in two directions. So basically in prokaryotes it just starts in one spot and then it just keeps going until both those lines have formed a new line, basically. So they split and then each one will form a new one. So prokaryotes, it's pretty simple. However, in eukaryotic chromosomes, our DNA replication is going to occur at hundreds of different places because it's more complex. It's going to take more to replicate it, and replication will proceed in both directions until each chromosome is completely copied. So it's not going to stop until our entire chromosome is copied. And the sites where separation and replication occur are called replication forks. So again, we have a double helix, and in order for us to copy our DNA, that double helix has to split apart. And so where that separation starts and then where our replication, making a new one, begins is called replication forks. And then duplicating DNA, so again, that's just kind of what I've said we've been doing. So before a cell divides, it duplicates its DNA in a process called replication, just making a new copy of it. And replication ensures that each resulting cell will have a complete set of DNA. So we need to do this, like probably around the mitosis time, right before then, to make sure our new cells are going to have, they will each have their own set of DNA. And then during DNA replication, the DNA molecule separates into two strands, then it produces two new complementary strands following the rules of base pairing. Then each strand of the double helix of DNA serves as a template for the new strand. So that's I've kind of broken this down step by step. So basically, the double helix will separate. So those are our complementary strands here. And so then each one of those new strands, or each one of those strands after it has been separated, will form a new strand to go along with it. So we'll have two new double helices after we start with just one. And then this is kind of a picture of what it looks like. So if we go on our far left here, we have that replication fork. So that's where it broke off, and you can see now where it broke off. And then if you look now, we have blue is our original strand of DNA, and now we have red that is replacing around it, forming our two new strands of DNA and where it's going to happen. Again, this might make a little more sense when we actually talk about it in class, but basically all that's going on here is the far left is our original strand, then it breaks at the replication fork, and then each one of those broken strands gets a new strand formed on top of it, with the A's and the T's and the C's and the G's matching up. And how this occurs, DNA replication is carried out by enzymes that unzip a molecule of DNA. So again, that's that going on right there, the unzipping. And then the hydrogen bonds between the base pairs are broken and the two strands of DNA unwind. Again, that's what I've been saying this whole time. Another word for it coming apart is unzipping, because it is almost like a zipper with the teeth lining up together. And our principal enzyme that's involved in DNA replication is called DNA polymerase. So this is a very important word that we need to know. This is an enzyme that's it's basically responsible for unzipping our DNA. And DNA polymerase will join individual nucleotides to produce a DNA molecule, and then it proofreads each new DNA strand. So that's our function there. If you have any questions, let me know.